Hi, everyone. Hi, I'm Harriet Sachs, volunteer for the APBD Research Foundation. I'm tonight's patient chat moderator, and I want to welcome everyone to the Zoom room. We have a full house, and that is just great. Our webinar, APBD Genes, Enzymes, and Research, oh my, is proudly sponsored by GeneDx. They were founded in the year 2000 by scientists from the National Institutes of Health. GeneDx's mission is to make genetic testing accessible for all patients with rare diseases. I need to tell you that the information presented in our webinar represents the views and opinions of our presenter and does not necessarily represent the views and opinions of GeneDx. And as always, our chats are not meant to be a substitute for medical advice from your healthcare team. I'm pleased to introduce you to Noelle Lynch, the newest addition to our foundation's team. Noelle is a genetic counseling grad student from Columbia University. She was inspired to enter the field by a desire to give sensitive care to patients who are receiving difficult news and experiencing long diagnostic odysseys. Noelle, the video platform is yours. Take it away.
Thank you, Harriet, for that lovely introduction. And thank you all for joining this webinar today. I am excited to speak with you, and I'd just like to thank the APBD Research Foundation as well as GeneDx for giving me this time to talk to you about APBD genetics, genes, enzymes, and research. Oh my. So just to kind of go over what we'll be talking about today, we'll do a quick genetics refresh about the genetics of APBD and just genetics in general. Then we'll talk more about the GBE1 enzyme. And then from there, we'll go into a discussion about the therapies that are currently being researched and use our background knowledge from the genetics research and from what we learned in the GBE1 enzyme segment. So we'll start off with that genetics. In order to understand the genetics of APBD, you have to understand what a gene is. So genes are the instruction manuals for everything in our body, from how we develop to how we function, how our organs function, and even how we grow. So we have over 20,000 genes, each of which are in charge of important bodily functions and do different jobs throughout the body. So how does this actually work? How do genes do this? So first, we start off with our instruction manuals. These are our genes. They're made of DNA. These instruction manuals get turned into messengers. So these messengers are able to deliver the information in order to turn that information into a protein. So the instruction manual becomes a messenger. Once that information is delivered, it's turned into a protein. And these proteins are the builders, they're the workers, or maybe even the building materials in our body in order to allow our body to actually achieve the growth and function and development that it needs to do. So what causes APBD? Where does it happen in this pathway? So what I'll start off with is how the pathway normally functions. So the gene that's associated with APBD is the GBE1 gene. I hope you can see my little mouse, but I'm circling the GBE1 gene, and this is our instruction manuals, our DNA. That gets turned into the messenger again, which then gets turned into the GBE1 enzyme. That's the protein that it gets turned into. Now, when someone has a change in one of these genes that makes it so that it's not working properly, they're called a carrier. They still have one copy of the GBE1 gene that is still working, it's still making the messenger, and it's still making plenty of the GBE1 enzyme. However, when someone has a change in both copies of their GBE1 gene that makes it so they can no longer work properly, they're not able to make enough of the messenger, and therefore that information isn't getting delivered, and it's not getting turned into the GBE1 enzyme. So they also don't have enough of this enzyme. And I just really want to highlight that individuals with APBD who don't have a functioning copy of GBE1 gene still have a little bit of this GBE1 enzyme. There's still a little bit that's getting made. It's just not enough for what the body needs. So I wanted to have a little audience poll. Has anyone explained the GBE1 enzyme's job to you? And our answers are yes, no, and I'm not sure.
All right, so at this point, everyone should see the question for the poll. Feel free to select an answer. Again, it is, has anyone explained the GBE1 enzyme's job to you? Looks like we got about half the folks have participated. I'm going to give a few more seconds for others. All right, I'm going to go ahead and close that poll. Noelle, you seeing
Okay.
those results?
Yeah, I am. That's exciting. Almost half of everyone has had the GBE1 enzymes job explained to them. And it looks like there's some people who haven't, and hopefully we'll discuss it, and everyone who has or hasn't heard about it will learn something. But it's exciting to see that about half of you know at least a little bit or more. Some people are not sure. So let's go ahead and talk about it as a group. In order to understand the GBE1 enzyme's job, we have to know what an enzyme is, right? So enzymes are a special type of protein that perform reactions in the body. So these are the workers that go throughout the body in order to do a specific job. Or sometimes they might be just located in a specific part of the body or the cell rather than throughout the entire body. They may even work together with other proteins or enzymes in order to get that job done. So I've just included some little graphics to help us understand what kinds of jobs it could do. So it might cut certain things, it might make things go faster, and they might make things get put together. So the enzyme that's associated with APBD is the GBE1 enzyme. And this enzyme works together with another enzyme called GYS. And they work as a team in order to form glycogen. Glycogen is our body's way of storing sugar. And this is especially important because sugar is very important for our brain. So we need to be able to store it, and we need to be able to break it down and use it when we need the sugar to be the energy for our brain. So GYS's job, if you look down here, this is our chain of sugars that are all getting added to a chain to become glycogen. So each of these little circles is our sugar, and then this whole chain is the glycogen that's being formed. So GYS adds to this chain. It adds sugars to the glycogen. Hopefully you saw that little animation. And then GBE1 comes along and cuts that chain once it gets too long. Then it sticks it back on the chain to form these nice little branches. So the way that we can remember what these enzymes are doing is that GBE1 stands for glycogen branching enzyme. So it's this enzyme that comes along and it makes a branch in the glycogen. GYS stands for glycogen synthase, GYS, with synthase meaning synthesis or to put together. So GYS is adding to the long chains of glycogen. The reason why they work on this team and the outcome that comes from it is this beautiful glycogen molecule with all these lovely little branches. It's actually 3D, so you can think of it almost like the top of a dandelion when it's all fluffy. Because it's got these branches off of these long chains in a spherical sort of way. And this is what they're able to create when GYS and GBE1 are in balance, when they're working together in order to create glycogen. But what happens in APBD? When we have very little amounts of the GBE1 enzyme, like I described earlier, the balance between GYS and GBE1 is thrown off. So GYS continues to add sugars onto the chain of glycogen without GBE1 creating all of these branches needed to create that complex 3D structure that I was telling you about. And instead, the glycogen looks like this, where it's very long chains with very few branches. So why are these long chains with few branches actually a problem? This poorly branched glycogen begins to accumulate over time, and it becomes large polyglucosan bodies. So that's the APB, right? The PB is the polyglucosan body. And you can imagine if you have these really long chains of glycogen, they're almost like a bunch of balls of yarn. And so if you have a bunch of string, a bunch of yarn that's all sitting together in a bunch, and you kind of start digging around in it, it's going to get tangled up. And pretty soon you're going to have this whole bunch of yarn that's all stuck together. And no matter what you do to try and detangle it, it's just going to be like that forever. It's going to be so knotted and you're not going to know what to do with it. That's kind of the same way that these polyglucosan bodies are created and then start to accumulate in the body. And specifically, they accumulate in the central and peripheral nervous system. So they accumulate in the central, being the brain and the spinal cord, as well as the peripheral nervous system, which is the nerves coming from the spinal cord. And what they do is they plug these nerves, just like this, preventing the movement of signals and nutrients to the other nerve cells. So if we look at this little sink with a plug of polyglucosan bodies, all of these nutrients and signals aren't able to make it to the rest of the body because it's plugged. And without those nutrients and signals, the cells that are over here are going to start to die off. And that's what results in the symptoms of APBD. So I have another audience poll. I wanted to know if you have seen a genetic counselor to have your genetic testing results explained to you. And the options are yes, no, or I'm not sure.
All right, everyone. So we got another poll on the screen. We've got about 3/4 of folks have voted. I'm going to give you 3 more seconds. All right, 3, 2, 1. All right.
Okay, 50-50. It's surprising to me as a genetic counseling student, but I have another question for you as well. Would you have wanted an additional discussion, or would you wanted a discussion with the genetic counselor at all in order to discuss the meaning of your genetic testing results? And this one's just a yes or no. And I know I'm a genetic counseling. Oh, sorry, Kalai.
I was gonna say, all right, got over 80% participation. I think we're ready to, 87 gets higher every time. All right, folks, I'm going to go ahead and close the poll so we can see the results. There we go.
I was going to say, I hope that you don't feel pressured to say yes, just because I am a genetic counseling student. I won't be upset if you say no. So 80% would have wanted an additional discussion, or if you had answered no to the first one, maybe you would have wanted a discussion with a genetic counselor at all. So hopefully this talk gives you some of the information that you might have gotten from a genetic counseling appointment. But there are genetic counselors who would also be happy to speak with you, I'm sure. So as we move on from talking about the genetics and the enzymes, now we can begin to understand some of the therapies that are being researched currently. And I wanted to add this disclaimer that I'm not personally involved in this research, and I don't have knowledge about timelines for treatment development. And really, this information is just to help you understand the underlying function of some of the therapies that are currently being researched. So what are these therapies? A few of the main ones, and the ones that I'll be discussing today, are gene therapy, enzyme replacement therapy, or ERT as it might be known, antisense oligonucleotides, or ASOs, which is what I'll call them because antisense oligonucleotides is a bit of a mouthful, and small molecules. And what is the goal of these therapies? It's going back to the balance of GYS and GBE1. We want them to be in balance again so that they can create these 3D glycogen structures that are really important, that are much easier for the body to store and to break down again. And in order to achieve this balance, there's kind of two ways that we can go about it. We can either increase the amount of GBE1, or what's actually easier sometimes in therapies is to decrease GYS, is to decrease something. So I'll walk through each of the types of therapies and what their goal is with this balance. So starting with gene therapy, the goal of gene therapy is to increase the amount of GBE1. So if we go back to our APBD model, there aren't any working copies of the GBE1 gene. And so there's not enough of this messenger being made and not enough of the GBE1 enzyme being made. Gene therapy inserts a working copy of the GBE1 gene. With this working copy, we can start to create more of that messenger again, which in turn makes more of the GBE1 enzyme and achieves that balance. And how is this done? How do you get a new gene? This is a diagram from Dr. Manassian, who is actually one of the researchers looking into gene therapy for APBD. And what they do is they insert the missing GBE1 gene into a virus. And I know we typically think of viruses as making us sick, but this is a specific kind of virus that has evolved with humans for a very long time and doesn't make us sick. But we know that viruses have this really unique function where they can get into the cells and they can inject things into human cells very efficiently. So they're really good for using in this therapy. So what they do is they then package up this virus with the GBE1 gene, and they deliver it through the spinal cord to the brain, with the hopes of getting it to as many cells in the brain as possible. And that's what they need to do in order for this therapy to be effective. So their research shows that currently they can reach about 20% of the cells in the brain, but they're hoping that with further testing and research that hopefully we could reach more. On to the next type of therapy. Enzyme replacement therapy is also working to increase the amount of GBE1 enzyme. And so again, if we go back to this diagram where we don't have the GBE1 gene working, so we're not making the messenger and not making the enzyme, it's just taking that GBE1 enzyme from an outside source, so from cells in a dish or perhaps from an animal source, and injecting them into a patient's body. So we're not changing anything about their genes or their messengers, we're just giving them more of that enzyme from a different source. And then that way, they're able to increase the GBE1 enzyme and achieve that balance. ASOs, or antisense oligonucleotides, work to decrease GYS. So this is the opposite side of our little equation. It's trying to decrease GYS, but it's not affecting the amount of GBE1. And I want to say that there are many different kinds of ASOs. They might have different types of goals, so this is just one kind. And what ASOs target is the messenger. So this type of ASO would target the messenger of GYS, encoding it, telling the body that this messenger needs to be destroyed. So the body goes in and it gets rid of the messenger so that information doesn't end up getting delivered. And the body makes less of the GYS enzyme, decreasing the amount of it and achieving balance with the GBE1 enzyme. Small molecules can also be used to decrease the amount of GYS. And small molecules, instead of targeting the messenger, they're used to target specific proteins or enzymes. And just like ASOs, there are many different kinds of small molecules. So this is just one of the many ways that they're being investigated, one of the many ways they could potentially be used. So in order to decrease GYS and achieve that balance, a small molecule could block GYS from being able to add any more sugars to the chain of glycogen. By doing this, we're not actually reducing the amount of GYS, but we're reducing its activity. So even though we're not reducing the amount of it, we're still achieving that balance because it can no longer do its job, it can no longer add sugars to the chain, and it's restoring that balance with GBE1. And this is a lovely little diagram from the APBD Research Foundation with some of the therapies that are being investigated, the programs and the partners that are investigating these different types of therapies, and how far along they are in their process. So hopefully now you should be able to recognize some of the names of these therapies. So small molecule, gene therapy, antisense oligonucleotide, gene therapy, small molecules again. This one says biologic, but you can look over here and says ERT. So this is enzyme replacement therapy. And the only one that we didn't discuss is this antibody enzyme fusion. And that's simply because I don't understand this one very well. And so I wouldn't be able to teach you guys about it very well. Over here, you can see where along the process they are with these types of therapies. So discovery is when they're looking into a type of therapy, they're looking into what is the chances that this could actually be helpful. So they're researching the proteins or the molecules. They might be using computer models to see if it could potentially work. And then from there, if they think that this is a valid type of therapy that could help, then they move into preclinical research. Preclinical research is when they start developing and testing these therapies to use on cells or to use in animals. And then based off of that research, they may or may not move into clinical trials, which is when those therapies would be tested in people. And I did want to highlight GHF201, most recently was approved for a compassionate care study in Israel, where it's been used in 3 individuals. And based off of that, it's now received orphan drug status by the FDA in the US. So that's our latest update about GHF201. There are currently no therapies that are in clinical trials, but I'm sure the APBD Research Foundation will let you know if they hear any news about that. So I know that we've talked about a lot of information, but if you wanted to learn more about the APBD genetics or more about some types of genetic counseling topics, you could go to the APBD Research Foundation YouTube and follow these steps through our playlist, the patient chat playlist, etc. But I'll be putting these links into our chat button after I'm done speaking, so you don't have to write this down or anything. And now that we've discussed the enzymes and some of the therapies being researched, and you have some of that background knowledge, you could also hear more about the current research on therapies from this recorded lecture from the 2022 APBD Scientific Community Conference, where you could actually hear from the researchers themselves discussing these therapies as well. And I'll also put this link into the chat. And I wanted to thank you so much for listening to me and hearing about all these exciting topics and what questions you have. And I will stop sharing my screen so I can see all your beautiful faces if you want to show them.
Thank you so much, Noelle. We're waiting for folks to post their questions in either the chat or go off mute, and I can call on you or raise your hand. And maybe while we're waiting for folks, Harriet, I know you probably have some questions fielded to you in advance if you want to start with one.
Okay, while you're doing that, let me go ahead and this came to me by email during this, so I want full credit for multitasking here. Okay, so here it reads: we have 23 pairs of chromosomes. Are chromosomes made of genes?
Is
Is
that the
that right?
question? That is an
Well, so far,
excellent
that's a
question.
beginning.
Mhm. Okay, well, I can
Okay.
answer that so far. So we do have 23 pairs of chromosomes. So we have 46 chromosomes in total. They're pairs of each other. So you're right, we have 23 pairs. And these are the structures that hold all of our DNA. So these are like libraries for all of the instruction manuals. And so in these libraries are our genes. Those are the instruction manuals and all of the bookshelves, all of the chromosomes. Not all of that information is actual genes. You could think of it as just empty pages on the bookshelf. But yes, our genes are located on there and they're all made of DNA, which is what makes up our chromosomes. I hope that answers the first part of the question.
Well, it does, because then the follow-on question was, where does DNA come into the picture? And what you just said is that the genes are made of DNA. Is that right?
Yes, so our DNA is like the language that makes up the instruction manuals, but instead of A through Z, it's A, T, G, C. There's only 4 letters, but that makes up the entire language of our genetic information, of the instruction manuals. And so they're basically just the letters or the molecules. That's how our body actually is able to interpret and is able to read through them. So our genes and our chromosomes are all made of DNA.
Thank you, Harriet. And Larry, I saw you just went off mute. Do you want to ask your question verbally?
Sure, I'd be happy to. I've a little bit familiar with gene editing, reading some things about like CRISPR technology. Do you know if anyone is doing any kind of research into whether that might provide an eventual treatment?
I don't know, based off of the 2022 kind of meeting and presentation of the research, I didn't see anything with CRISPR from that conference. Harriet, I don't know if you've heard of CRISPR being investigated yet.
I am not aware of CRISPR being investigated, mainly because there seems to be some issues with the cut. CRISPR means cutting into the gene. Make sure I'm saying this right, or you say it. Don't, I'm not there to instruct, but it's a cutting activity. And something about the cutting is not always reliable. Is that what the problem is?
I think there could be a lot of different complications when it comes to CRISPR. As far as I know so far, CRISPR is really useful for conditions that often have the same genetic change that are causing the condition, because CRISPR is very specific. So it's looking for a specific change that it can cut out and then it can replace. But a lot of people with APBD might have different genetic changes that are causing that gene no longer to function, because there can be lots of different changes in lots of different areas within that instruction manual that make it so that it can't function properly. So if everyone had the same genetic change, CRISPR would be a really great option. But unfortunately, lots of people have very rare ones. And so maybe only one person could be treated with that CRISPR, and then they would have to design a new one. But that's just one complication that comes to mind. Does that answer your question?
Yes, it does. Thank you.
Thank you. We have a question in the chat, and then I'm going to go to Kathy. The question in the chat is, do any of the current therapies unclog the drain?
That is a great question. So currently, as far as I know, the therapies aren't undoing anything, but they're trying to prevent more of those polyglucosan bodies from accumulating. But I think as of right now, there's none that they have figured out to actually go in there and unclog the drain or try and, pull apart those polyglucosan bodies.
Thank you. We have a
I'd
question.
like to add, though,
oh,
that
great.
there are other disorders that have polyglucosan bodies involved. We don't have the corner on the market at APBD. And there are some researchers who are looking at basically dissolving the polyglucosan bodies for these other diseases. And the idea is, if it works there, can it work for us as well? So it's not totally bad news. It's, there's something going on in somebody else's backyard that we may find very valuable.
Thank you for adding that, Harriet.
Thank you.
Thank you, Harriet. Kathy, you're up next.
Okay, thank you. Do you understand why some people progress more rapidly?
I think at this point in genetics, we aren't completely sure why different conditions affect people differently. And we can kind of think of this like the cold. Why do some people get a cold and get a stuffy nose, while some people get a sore throat, while some people get a cold and end up in the hospital? And so there are just different parts of us, maybe other genetics, maybe other health conditions that can contribute to why someone might have different symptoms or might have progression of symptoms faster than someone else.
Great, Harriet. We have a chance for you to take on another group question.
Okay. So our listeners heard the thing about 20%. Can you repeat that? What does the 20% refer to?
Yes. So if we go back to gene therapy, right? So our gene is getting put into a virus, and that virus is getting packaged and delivered to the brain. And what we want that virus to do is to insert that gene into as many cells in the brain as it can. And right now, according to Dr. Manassian, they can reach about 20% of the cells. So that virus is able to get the working gene, the working GBE1 gene, into 20% of the cells in the brain. So 80% still don't have the functioning GBE1 gene and still aren't making that enzyme, the GBE1 enzyme, whereas 20% are. But ideally, that number would be much higher. We want it to reach as many cells in the brain as possible. So that's what they are continuing to research.
But does 20% do enough, or they really do have to up the number? This is Harriet's question. Do they really have to up the number before it's something that we could start partying over?
It sounded like, I'm not an expert on this. Based off of what Dr. Manassian said in the lecture, it sounded like it needed to be increased, and that wasn't as much as they were hoping for, not as much as they were expecting. And if we think back to our carriers who don't have any symptoms of the condition, they have one working copy of the GBE1 gene and one non-working copy. So we'd think that they'd be at about 50% of functioning GBE1 enzyme to someone who had two working copies. So is 50% the magic number? We don't know yet.
We have a question in the chat. What specifically is causing myelin damage in the process?
So those, the myelin cells are part of the nerves. And so we can think of it similarly to the plug. And that's the signals and the nutrients not getting to those cells. And so the plug is blocking it. The polyglucosan bodies are preventing the signals and the nutrients from getting to those myelin cells that are around the nerves. Was that specific enough?
We'll see if there's a follow-up question there. Larry, did you have another question? I wanted to make sure I didn't miss you.
I just had a thought, and I don't know if this is Noelle's expertise or not, but why is it that most of us, I think, start with lower extremity neuropathy and not upper extremity neuropathy.
I think you're right that that's not necessarily my area of expertise, but I do think that is an excellent question that I'm sure someone out there knows, just not me.
Great
Great.
question. I wish I knew the answer too.
Yeah.
And I have a quarter for anybody who comes up with the answer. Okay.
Harriet, we can go to you for another crowdsourced question.
Okay. Should my adult child get tested for APBD? Obviously, we know this is going to be a carrier.
Mhm.
Will it do me any, will it do our patients any good? Does it do anybody any good?
I am maybe biased because I am a genetic counseling student, and I do think that genetic testing has functionality. And the reason why it can be helpful for someone to get genetic testing done is because then we know that they're going to be a carrier, but then that report is in their medical chart and their doctors know that they're a carrier. And if they plan on having kids, for example, that's in their chart and something that, for example, an OB might be able to see and could trigger that conversation about carrier screening for their partners. Because we would also want to check and do genetic testing on their partners to see if they were also carriers of APBD. And if someone doesn't have that somewhere in their medical record, that's easy for a provider to take note of. These types of things, unfortunately, can sometimes be forgotten about.
Great. It looks like, Harriet, we can go to you again for another question.
Hey, so you said that there are this is Harriet's question. You said that there are many different kinds of mutations that our patients can be possessing. So we know that some are more common than others, but do you have any idea how many the many is?
I don't have any idea of how many could be, but for a lot of conditions, especially conditions that are common in the Ashkenazi Jewish population, there can be certain types of changes that are more common. So exact changes that are more common and are just seen more common in people with certain ancestries or ethnicities. But then at the same time, there can be very, very rare changes where maybe it's only in one person. So it can be difficult to estimate exactly how many many is. I might have overestimated by saying many, but we'll just say many.
Great. All right,
It
folks.
seems really amazing that especially if the APBD patient is not of Ashkenazi Jewish ancestry, what are the odds that the parents found each other to create the non-Ashkenazi Jewish? It just seems almost impossible unless those mutations are out there in huge numbers and we just don't realize it.
I think that's the question for so many rare conditions, is just how did these star-crossed lovers find each other? And it was just such a rare event. And I think it's something that genetics is going to be continuing to research, just trying to do more genetic testing on people and trying to see what do we all carry? How common are these things? How common is it to be a carrier? Because that can give us a lot of information about the condition too.
Thank you, Noelle. Do we have any other questions in our chat or on the floor, Calliope?
No, I just got one in. Is the data on all offsprings inheriting the double non-working GBE gene? Oh, I guess it's the question. Is there data on offsprings inheriting the double non-working GBE gene incidence? I've seen that siblings frequently inherit the two from carrier parents.
Okay, this is a great question. So this kind of goes back to, I think, the other genetics talk that I put the link in the chat does a really great job of explaining inheritance with APBD and why we know all the children of people with APBD are carriers, but I can go over it briefly. So we each have 2 copies of every single gene in our body, including the GBE1 gene, the gene associated with APBD. We inherit 1 copy from our mom, and we inherit 1 copy from our dad. So individuals who are carriers have one working copy and one non-working copy. When two carriers have children together, if they only pass on, so if mom passes on the non-working copy and then dad passes on the non-working copy, this child will develop APBD, as far as we know. And so that's about a 1/4 chance, right? If there's two carrier parents who have a child, there's a 1/4 chance that that child will inherit only those non-working copies. Now, when this individual goes on to have children, we're assuming for the most part that they had children with someone who was not a carrier, because it's very rare to be a carrier of APBD, right? And so when they have a child, they only can pass on the non-working copy. So it doesn't matter which one they pass on because both of them aren't working. So we know, no matter what, that their child is going to be a carrier of APBD. Now, the only chance that it would be for their child to, the child of someone with APBD, to inherit APBD was if the other parent was also a carrier, which is pretty rare. Does that answer the question? So we know that all children of individuals with APBD will inherit one change from their parent as APBD and will be carriers, at least.
Great, thank you. It looks like Kathy has another question.
Yes, I wanted to just share my experience. I have a daughter who's 37 now. And after I was diagnosed, I was in touch with Columbia, and she wanted to be tested when she heard when she looked up what the disease meant. And I spoke to Professor Ackman, and he suggested that my husband be tested because, and I think I spoke to other people in our foundation, and they suggested that just test my husband. And because the concern is that, as we know, with medical records today and computers, there are no secrets anymore. And the reason why they didn't want her tested is if she was looking for long-term care disability, that she could get refused because of her past history. She has no symptoms of it. And so we tested my husband and he was negative. So we were at least relieved that she will not get what happened to me or my brother, and that was reassuring. And that stopped us from causing any further questions or concerns. Thank you.
I think that's an excellent point, Kathy, that we would only be concerned of a child with someone who has APBD also having the condition if their other parent was a carrier of the condition, right? So we know they're at least going to be a carrier, but if the other parent was a carrier, then their child might be at risk of APBD. So testing the other parent, testing your husband makes a lot of sense because if he's not a carrier, then we know that your children were not at risk of inheriting the condition. But if we just go immediately to test your children when they don't have any symptoms of the condition and they're younger, then you're right, there can be insurance issues. So I think this can be helpful. There is a law called the Genetic Information Non-Discrimination Act. It's called GINA. It means that your genetic information and your health history cannot be used against you for employment or health insurance. But you are exactly right, Kathy. It can be used against you for long-term care and disability, as well as life insurance, right? And could be used to either deny someone coverage or to increase premiums. So it definitely makes sense that they recommended you test your husband first before testing your daughter.
Great,
Well,
thank you.
you have done a remarkable job. You put together a fantastic presentation. It really helps. Pictures are worth many, many words, and it really did help. Thank you so much for the time that you put in to create that for us, and then the time you gave us this evening to share it with us, and then to answer our questions off the wall. just terrific, and we're so grateful for all that you've given us. I look forward to having more work for you, with you, as you do other projects for our foundation. I thank you again on behalf of everybody in the Zoom room.